Hello, my name is Caitlin McCann and I'll be today's moderator. This webinar, sponsored by QIAGEN Digital Insights, is titled Longitudinal Mutational Landscape and Transcriptomic Analysis of Aggressive Uterine Leiomyosarcoma in a Patient with Exceptional Survival. The panelists for today's webinar include Dr. Tatiana Omelchenko, a senior research scientist at Rockefeller University, Deep Pandya, a research associate at Danbury Hospital, and Dr. Jean-Noel Billaud, senior principal scientist at QIAGEN Digital Insights. And with that, I'll turn it over to Dr. Omelchenko. Go ahead.
Hello, everyone. I would like to thank organizers of this webinar for the opportunity to show our work on the rare and deadly metastatic cancer called Leiomyosarcoma. This is very aggressive mesenchymal cancer that accounts only for 10% to 20% of the old soft tissue sarcomas. So leiomyosarcoma arises from the smooth muscle cells of different structures and organs. And today I will show you data on the leiomyosarcoma of the uterus that is often diagnosed at the metastatic stage of the disease and has no effective treatment protocol. So we need to do hard work to understand molecular mechanisms and find treatment. I would like to start with the target organ, the uterus. This is a specialized organ with the primary function to provide the best environment for the embryo to implant and to gestate to full term. And as you can see here, on the left panel, the uterine wall is thick, and it is composed of the smooth muscle bundles composed of the smooth muscle cells. So from the one side, from the outside, is covered by the perimetrium. It's a serous tissue. And on the other side, internal side, is covered with the endometrium. So here you can see on the right side, the zoom view of the uterine wall and its structure. So the endometrium is the mucosal wall, which is the focus of most many studies on gynecological cancer of endometrial cancer specifically. So the essential function of the smooth cell cells is contraction. And they are enriched in the cytoskeleton, which is responsible for cytoskeletal elements, which are responsible for the contraction. But these elements are not specific to the leiomyosarcoma, unfortunately, so we cannot use them as specific markers. So one of the features of the smooth muscle cells is their quiescence. So these cells not proliferating very often, but during inflammation or cancer, they start proliferate. And due to low proliferation rate of the smooth muscle cells, of course, uterine leiomyosarcoma is a very rare malignancy. So in comparison with its benign smooth muscle tumor leiomyoma, which you can see on the left side, the uterine leiomyosarcoma on the right side show very biologically aggressive behavior leading to the invasion and local and distant metastases. And morphologically, it shows nuclear ITP and high mitotic activity. The prognosis of this disease is very poor, and there is a very high recurrence rate of the disease even after complete removal of the uterus. And as I mentioned earlier, the 5-year survival rate is very poor. So analyzing the molecular mechanisms of what causes the disease, it's important to look at the hereditary diseases. And fortunately for us, we can learn that hereditary retinoblastoma patients, they have a higher chance to develop a leiomyosarcoma, including uterine leiomyosarcoma. Patients with the Lee-Fraumenti syndrome, which have inherited mutation in TP53 gene, also have a chance to develop leiomyosarcoma. In addition to that, there are some external factors like radiation and tamoxifen exposure, which can also lead to uterine leiomyosarcoma malignancy. So as you can see here on prognostic factors, the very late stage, which is metastatic stage 4, the survival rate is very low. And how I would like to cite Alessandra Santin, the team leader at Yale University, who published this year a very good article, very interesting. So he said that we have no cure, so uLMS is still incurable, especially the advanced metastatic disease. So a limited number of studies have evaluated the genetic landscape of the uterine leiomyosarcoma. And they revealed that uLMS is very highly heterogeneous, and with their complex copy number alterations and distinct losses of tumor suppressor genes without known recurrent activating mutations. And the most prominent example is the loss of the RB1 retinoblastoma protein, as well as the PTEN. So in addition to that, the other at least few very pronounced genes which are mutated are TP53, RB1, ITRX, BRCA, and MET12. And since we're focusing here on a specific pathway, I would like to mention work of Eva Hernanda from Carlos Cardon-Carda at Mount Sinai, who published very great study on the role of the PI3 kinase and AKT in development of leiomyosarcoma. So using mouse model, they performed knockout of PTEN in all smooth muscle cells and found development of leiomyosarcoma in multiple locations. And recent papers are also very promising in testing inhibitors against this axis of PI3 kinase, AKT-mTOR pathway. In addition to the other targets, inhibiting targets like BRCA1 and c-MYC. However, this knowledge is quite limited, so we are working hard to understand the molecular basis of this disease. And specifically, we had a very unique case where a patient had an exceptional survivor. And since 2014, we had this unique chance to collect samples over time of developing metastasis as well as the primary tumor and analyze the development of the disease and the future evolution of the tumor. So here on the schematic, you can see the time of the surgery labeled at the top, in the top row. And so we collected samples from primary tumor, metastasis lesions for A and B. And then preliminary, we had some data available from the ANCA panel done on the metastasis number 2 and impact a panel of about 400 genes on metastasis number 3. So we performed whole exome and transcriptome sequencing, and as well as established cell line and multiple PDX models from different metastases to understand the molecular nature and to find targets for the treatment of this specific case. So our objective was to propose first mechanisms and find treatment strategies so that this can be applied to other sarcomas with the same genomic landscape and find the treatment which is well tolerated, because as we know, chemotherapy is affecting multiple organs, not only tumor itself. So we're looking really for metabolic derived treatment, and we're looking forward to it. So this is the experimental procedure describing details of our protocol. So we are first assessing genomic alterations and copy number profile using whole genome sequencing, as well as expression of genes using transcriptome sequencing. And also, we are not showing here, but we are evaluating also tumor mutation burden data, and we put it in the context of the available data to understand which patterns are present and what would be the best drug target. So now I'm moving to DEEP, and DEEP will tell more specifically on the flow of the mutational landscape analysis. Thank you.
Hi, this is Pandya. Thank you, Tatiana, and thank you, Christy, for introducing me, and Tatiana for introducing the clinical part of the study. So as Tatiana mentioned that we have a 55-year-old female with stage 4 metastatic uLMS that we were lucky to follow for last 7 years and be able to collect 1 primary and 4 metastatic sample as shown in the table. We were also able to collect 2 normal adjacent tissue from the metastatic 4 surgery. So here is the workflow, which was very well crafted and validated multiple times using various tools offered by QIAGEN, such as QIAGEN CLC Genomics, Clinical Insight, and IPA. And they were all the data were run at the Nuance Health Danbury Hospital facility, where I work as a bioinformatician. So on the left side, there is a workflow for whole exome sequencing. And this whole exome sequencing, we use low frequency variant detection tool to identify mutation for each sample and among the sample, but we also use whole exome sequencing for prediction of CNV in each sample and among the sample as well. Whole exome sequencing was performed at 250 to 400x DAB to get even the low frequency variant with a high confidence. Once all the CNV and variant were produced, we process through QIAGEN's Clinical Insight, which will annotate and filter the variant to assign the functional impact. On the right is the RNA-seq workflow. We aim to produce 110 million reads, which basically will cover even the low expressed genes. In addition, we also produce a mutation from RNA, so we can correlate that with the DNA mutation. At the end, DSeq-based differential expression analysis was performed between metastatic sample and primary. And then we combine, this is a unique way of analyzing data where we combine mutation with the expression profile, and then we uploaded that data set to the Ingenuity pathway analysis to predict canonical pathway, upstream regulator, and at the end, some potential drug target. So we aim not to go in one direction to analyze the progression of the disease. We try to integrate a multiple workflow, multiple analyte to get the answer of the tumor progression more in a tumor biology rather than just progression of the disease and looking at one directional biomarker. So that being said, let me talk about some basic workflow for transcriptomic and whole exome analysis, because this presentation is in work in progress. So we also want to introduce the approach we use to reach to some conclusion in the tumor biology. So for transcriptomic workflow, this is the simplified workflow shown in the slide, where using this workflow with the CLC Genomics workbench, we produce not just gene expression, we use transcript expression as well as RNA mutation, as I mentioned earlier. We also produce a fusion gene, which we fusion genes using this transcriptomic workflow, which we will not talk about today. HG38 and Ensemble version 99 were used as a reference gene for gene track and mRNA and the genome track. So as you can see in the on the table left, in the red box, for most of the sample, we were able to get more than 110 million reads, and most of them were assigned to the, most of the pair reads were assigned to the genome. We went further and looked the distribution of a biotype, and we see that most of these reads were assigned to a protein coding region. Some of them are assigned to written intron and antisense RNA, but the percentage of those reads are very low. So that shows the efficiency of library prep and sequencing through Illumina HiSeq. I think it was sequenced on the NovaSeq platform. So before diving into the individual gene or group of gene which are differentially expressed, we just wanted to look at the overall profile of gene expression in each sample. And then when we clustered them together for the top 25 genes, it revealed the major network of primary, metastatic, and normal samples as shown in that heat map. We also did a PCA analysis, principal component analysis, on entire transcriptomic profile, which revealed that similar to the heat map, that the primary, metastatic, and normal sample are very distant from each other, which intrigued us to look even further. So we performed differential gene expression analysis first between primary and each metastatic sample, and then compare each of the comparison to obtain common signature of pattern that differ from a primary sample. We use very stringent cutoff of fold change of 3 and bone for a p-value of less than 0.05. We found that there are more than 600 common genes, which suggests that there are some common signature pattern in metastatic sample as compared to the primary, as well as there are some unique differentially expressed gene in each metastatic sample, which suggests that sample did not arise from each other, and it might arise from the primary sample. And before we further look into the gene expression profile and pathways and all other prediction, let me briefly introduce the workflow that we use for whole exome sequencing. So this is, again, the very simplified version of the workflow that has been used to process the whole exome sequencing using CLC genomics. This workflow basically are the major steps in this workflow are alignment, realignment, removing duplication reads, generation of a structural variant, indel, 4-pass low frequency variant detection. And based on the filter shown on the right, tool will generate variant with read as low as 2, which is very computational. It consumes a lot of time. And we try to get the allele frequency as low as 1%. But we apply those filters to get the high quality variant, not the false positive. All the variant files which are generated here, or all the variants which are generated in sample, were then processed through a QIAGEN clinical insight. But before that, just want to show the specificity of the assay. So any whole exome sequencing, you will look for the specificity of the reads on target. So as you can see, this is a chromosome-wise distribution, but overall for each sample, more than 200 million reads were produced, and more than 80% of specificity on the target were achieved for all the sample. In terms of the coverage, we got more than 200x coverage for most of the sample, except for a primary tumor that due to the nature of the sample, FFP tissue, and is still enough to call those low frequency variant. So we accepted that data and proceed further. And so as I mentioned, all variant files were then processed through QCI platform for filtering and annotation. So these, we applied a multiple filter which are available through QCI, like confidence filter to quality filter those variant. And as of now, we applied the 5% allele frequency cutoff and at least minimum 10 reads. For minor allele frequency in the public data set, we went and filtered those variant which has more than 0.1% minor allele frequency in the, so it has to be rare to be considered here. For a predicted deleterious filter, we consider through ACMG guideline whomever are pathogenic, likely pathogenic or uncertain. Or for with the other algorithm, there has to be frame shift in frame, then or some functional impact predicted through SIFT or polyphen. So we basically use a multiple algorithm to compute the classification, not just one. And genetic analysis filter is just applied to see how many are common between all the samples, or you want to select just one, then it will show you the unique variant in each sample. So at the end, we started looking at what are the shared variant between all samples, including one primary and 4 metastatic samples. So we found 79 share variant. When we try to find some correlation of this variant to leiomyosarcoma, we found 17 out of 79 have some association with the uLMS. Gray color show variant with uncertain significance, and green are being variant with some benign variant, but some functional impact. Although I just want to emphasize here that these annotation were given for the uLMS, and it's as Tatiana mentioned, it's not a very common disease, so not that much clinical significance or data available to actually consider them as a likely pathogenic or pathogenic. So, but how this computational annotation were given, I just want to show you one gene called phosphodiesterase 4D interacting protein. So that protein has given the uncertain significance at tier 3, but if you look at overall cancer, gene prevalence is found around 7%, and due to no clinical significance, the tier 3 were given to that variant. But at the same time, this variant is very rare in the common population, has a deleterious effect, so that can be considered as a likely pathogenic for overall cancer. So my point here is this uncertain significance and the benign or likely benign annotation might change if we have more data on the uLMS. But at this point, if we go back and consider just for a cancer, some of these uncertain significance variant can be considered as a likely pathogenic, and that shows that some of the variants can be interesting to even investigate it further. So we went further to analyze sample-specific unique variant in each sample, including primary, and we identify functional and pathogenic variant in genes such as IQGA4, that's a tumor suppressor, SAMD1 and CYP4F8, it's a member of cytochrome P450 superfamily of enzyme that is involved in a drug metabolism and synthesis of a lipid and cholesterol. But surprisingly, 2 oncogenic mutation which are described in uLMS before has been found in a metastatic sample 2, but not in the primary, in P10 and TP53. Metastatic 3 has produced 1 pathogenic variant, but other functional variant based on SIFT and polyphen with a high allele frequency. Metastatic 4 sample did not produce any pathogenic, but they still produce some functional variant. In the bottom, we manually created some of the gene from those 79, which carried from a primary tumor to the till the metastatic 4 samples. So we wanted to check the progression of the disease, and so we focus on those gene variant where either allele frequency increased or decreased from the time they identify in a primary tumor. So we found 5 gene variant in the gene ZG16B, FGF12, Tenurine M1, Plexin A4, and UNC5A. We will talk about those genes in the next few slides. But as you can see, the allele frequency of this variant from a primary to the metastatic 4 has increased. Some of them double, or some of them increase at least by 30%. For UNC5A, it increased in metastatic surgery 3, but for a 4, it decreased a little bit, but still more than a primary sample. But for the gene we just described earlier, PD4D, the allele frequency actually decreased from 80% to almost 36%. So I think this is very interesting. That shows some tumor progression, and it's hard to sometimes find one variant, but we found like almost 6 of them, which at least 5 of them where allele frequency increased, which kind of reflect the tumor progression. So let's have a look at those genes. The one gene called zymogen granule protein 16B. It's also called POF, pancreatic adenocarcinoma upregulated factor. The function for those genes are carbohydrate binding and glycosylation. It's not only described in pancreatic cancer, but these are in blue are notable publication where it is described, and one. It is also described as a prognostic biomarker in breast cancer, and other is silencing of this gene increase the sensitivity of pancreatic cancer cell to gemcitabine, which is one of the chemotherapy given for pancreatic cancer patient. FGF12 is more known to the audience, which is fibroblast growth factor 12. It is a member of FGF family. The function, there are a lot of functions. Some of them are mitogenic and cell survival activity, embryonic development, tumor growth, invasion, nervous system development, etc. Some associated diseases are development disorder, epilepsy, and also described in colon cancer. Some related known pathway are GPCR and PAC pathway, and it is shown that a mutation, a functional mutation in the promoter of this gene has an increased risk for bladder transitional cell carcinoma. So 10M1 is the tenurin transmembrane protein 1. The function is it is expressed in the neurons and may function as a cellular signal transducer. It is shown in liver cancer, hepatocellular carcinoma, and epithelial cancer. And again, overexpression of this is a potential marker for disease progression in a papillary thyroid carcinoma. We also kind of show a progression of increasing allele frequency. So it's very interesting gene to further investigate. Plexin-4 is slightly unknown. It's a co-receptor of SEMA3A. Related diseases are just Down syndrome and neoplasia. But again, it's shown to promote tumor progression and tumor angiogenesis by enhancement of VEGF and B-VEGF signaling. And we will see that VEGF signaling is activated in our data set when we talk about those pathways which are activated in our data set. UNC5A is natrin receptor A. It's also involved in exon guidance and natrin signaling. This is again a tumor suppressor gene, or it also target tumor suppressors such as P53 and regulate the apoptosis. So my point here is we found this gene which should be gene variant which should be further investigated for the progression of the disease, but we just don't want to limit ourselves looking at only mutation profile, although it is interesting. So we further went ahead, and from whole exome sequencing data, we started analyzing the data for copy number variation. So before we dive into our data, we generated C and V. Just want to emphasize that for the sample has been sent to the clinically available MSK impact panel after the metastatic surgery, and we saw that these are all copy number variation in TP53, RB, and P10 have been detected with a higher fold change, but very few mutations. So it correlates with the finding that soft tissue sarcoma is pretty much copy number variation disease and less mutation disease. So then we started looking into copy number variation profile of our own sample. But before I talk about our finding, I just want to talk about the algorithm that we use, which will interest some of the bioinformatician in the audience. This is it's basically this algorithm used by CLC Genomics Workbench are determined by normalized. So CNV here are determined by normalized fold change in average in the coverage in sample compared to control. As shown in the report figure on the left, tool will first identify chromosomal disproportion in the coverage. And in this case, we found disproportion in 1 and 19 as a gain and 11 with the loss. Basically, each target will be plotted on respective chromosome and ratio of coverage will be determined. Anything outside confidence interval will be considered gain or loss. So all the gene CNV which are predicted here will again filter and annotate with the QCI pipeline. And we generated region level, target level, and gene level CNV. But for today's talk, we are going to focus only on the gene level. So as we done for the variants, we first started looking into the common signature of CNV at the gene level, and same filtering as variant has been applied. So in total, we started with 7,000 CNV, and after all filtering, we were able to identify 299 CNV, which are shared between primary or all metastatic sample, and that is shown in the figure pretty much with the red boxes. So here I just want to emphasize that we not only identify those or validated those CNV which are predicted by a clinical panel offered by MSK, we were able to detect even further. So that kind of validate our sequencing, our bioinformatics analysis. But then we have advantage of whole exome sequencing. We are not limited to a panel, so we were able to detect even more CNV, like in the gene VRAP53 or TSC1. And these are the tire on the right side. As I'm not sure if you can see that, but on the right top of the box, you can see that there are annotation given like S or R. S is for sensitivity to therapy. R is for resistant to therapy. If tire 1 or 2 is given, that shows some strong clinical significance. Otherwise, tire 3 is for just unknown significance. So for uLMS, it is believed that most of them will be tire 3 because it's not very well studied disease. So this is just an example of, so as Tatiana mentioned, P10 has been described extensively for uLMS. And we detected loss of function CNV with at least we can assume that one copy was lost, because this is based on a fold chain. We cannot actually determine how many copies were lost through whole exome sequencing. We think that there is one copy lost through the algorithm, and there is the deletion happened on exon 1 to 9. And for P10, the tire 2C were given because there is some inclusion criteria for some clinical trial going on for this gene. And based on the rarity of the CNV and some functional impact studied in literature, pathogenic computational annotation was given. So we just didn't limit ourselves by looking at the common CNV which are predicted among each sample. We also tried to look for a unique CNV which are produced in each of them. And we actually find very interesting in metastatic 4 samples, CNV in BCOR, CDKN1B, and ATRX. There were no gain of function CNV identified in these samples. So then, just to predict some canonical pathway, upstream regulator, and drug targets, we use the tool offered by QIAGEN called Ingenuity Pathway Analysis, where we can actually integrate our mutation finding with those of the expression analysis and all the differential expression which were performed through CLC can be uploaded here, and that by using a very complex algorithm and Z-square cutoff, we were able to predict some of the pathway which are shown here. So basically, the comparison were done between a primary sample with the metastatic sample, as shown in the heat map, and we were able to predict, the IPA predicted that Hippo signaling, P10 and Rho GDI signaling are predicted to be inhibited in all 4 samples when compared to primary, and such important pathways such as mTOR signaling, PI3K signaling, VEGF signaling are all predicted to be activated. For today's presentation, I'll be focusing more on the mTOR pathway, as mTOR pathway has some overlap of genes between mTOR, P10 and PI3K. So it will cover more overview of our data than any other pathway. So here is the prediction of mTOR signaling pathway, which is activated by looking at the differential gene expression signature from our own data set. As you can see from the heat map on the right, pink color is for the upregulation of the gene, and green means the downregulation of the actual expression. Orange means the prediction of activation, and blue means prediction of inhibition. So as you can see here, that some of the important signaling like PI3K, VEGF, some complex mTOR, C1, mTOR, C2, all are activated. On the other side, TSC1 and TSC2 are predicted to be inhibited in the mTOR pathway. So, and just want to emphasize that the pattern of differential expression when compared to the primary sample has been consistent throughout all 4 comparison, which is shown in the heat map. So it's just not in metastatic 2. We are just showing the one of the as an example, metastatic 2 versus primary. So just to talk about mTOR pathway, principal pathway that signal through mTOR is the PI3K AKT signal transduction pathway, which is involved in cell survival and proliferation. The signal from activated receptor is transferred directly to PI3K AKT pathway, or alternatively through activated growth factor receptor that signal through oncogenic RAS, which is pivotal activator of MAPK pathway. So the major component of mTOR pathway are mTOR complex C1 and mTOR C2. mTOR C1 has mTOR, Raptor, and GBL. mTOR C2 has GBL, Richter, and mTOR. So as a central modulator of proliferative signal transduction, mTOR is the ideal therapeutic target against the cancer, and that's why we used IPA drug prediction tool, which will show us if there are any drugs which can target the major component of this pathway. And we found the combination of everolimus and gefitinib, and individual drug called dactolisib. We will describe this drug in the next slide, which can actually target some of the major component of mTOR to inhibit the activation. I just want to emphasize that with the mTOR and PI3K AKT signaling, there are 32 genes which overlap with each other, and with P10, there are 15 gene overlap. So about those drugs, gefitinib has been FDA approved for a non-small cell lung cancer. It can target EGFR, BCL2, VEGF. For everolimus, it can target directly, it binds mTOR. It is also approved for neuroendocrine lung cancer and some other cancer like renal cell carcinoma. On the other hand, dactolisib is a potential drug. I think it's studied in phase 2, phase 3. trial, but not been FDA approved for any cancer yet. So there is some potential advantage here to use for the uLMS. It also binds to mTOR. So dactolisib can bind to 6-component mTOR C1, mTOR C2, and PI3K, where the combo of gefitinib and everolimus can bind FKBP1, mTOR, mTOR C1, and mTOR C2 complex. So as we talked that we combine the signature of the mutation and the expression profile, and here I mentioned that TSC2 has been predicted to be inhibited, but we also found there is a mutation in TSC2, likely pathogenic mutation, which is a loss of function. So here the mutation, the prediction, and the prediction done by expression profile match with each other. So that that made us look further into the TSC2 network, and those genes which are responsible for prediction of inhibition of TSC2 show those genes are involved in invasion of cells and metastatic process, and the heat map of all these genes are throughout the metastatic samples as shown in the right. So that being said, this is one way of looking into and finding the therapeutic target, but we also wanted to focus our study on finding or comparing our data set and those upstream regulator and canonical pathway to a public data set. So for that reason, we used a platform called Analysis Match, and I would like to ask Dr. Jean-Noel because he's an expert on this tool, to shed some insight into this tool. So thank you from my end, Jean-Noel.
Thank you, Deep. So as Deep was mentioning, we are using features to look for potential treatments. So obviously everything we show is hypothetical and needs to be validated in the lab. But the features we use, it's called Analysis Match. And the principle is very simple. We have a bank of over 96 comparison expression data set that span oncological data set or other data set, inflammatory and you name it, neurological and so on. And we have the power to analyze each of these each of these comparisons and determine, like we have done for that Met4C that you see on the slide here, an upstream regulator, a canonical pathways, and biological processes. And we determine a Z-score. Based on the Z-score, we are able to see if there is what we call a match, which is represented by the magenta color in the bottom, or an anti-match, which is represented by the blue. The match means that some of the upstream regulator, for instance, that you see in the comparison shown on the bottom left of that slide will be shared with Met4C versus primary. In the blue means some of the upstream regulator that you find as well in the Met4C goes in the inverse directions. Right? So in other words, what you find in Met4Cs that is predicted to be activated, in the blue, you will find them inhibited. And the Met4C upstream regulator activated in a magenta, you will find them in a magenta as activated as well. Match, anti-match. So based on that principle and through a filtering scheme, we are able to narrow down to look for comparison expression that have a notion of the treatment versus control. And the treatment versus control will allow us to see at what point and which analysis will revert the data that we have obtained with the metastatic versus primaries. So I'm looking for blue. I'm looking for data sets that are dissimilar in terms of usage of the upstream regulators. And we have found a bunch of them. And I'm going to show you the heat map of these particular data sets. This is the heat map that on the left, you have all the upstream regulators. On the pink at the top of the cluster, this is our data set, the metastatic 4C in that case versus the primary. And you see basically blue and orange color. Blue means all these upstream regulators you see on the left are predicted to be inhibited in that metastatic 4C versus primary. And in orange, all the upstream regulators that are predicted to be activated. So the goal is when you narrow down to treatment versus control type of experiment, you would like to see at what point and what treatment can revert that particular pattern you see on the left. And we have come up with these hypothetical treatments. Obviously, it needs to be tested, but bortezomib, birabresib, gefitinib, and verifanimib are potential drugs that will revert the pattern you see on the far left for our metastatic. Everything in blue becomes orange, everything in orange becomes blue. But we have also looked at that at the canonical pathway level from that same 20 dataset comparison from analysis match. And this is the case here. So the same things, our dataset is in pink, the MET4C versus primary. In blue, all the pathways that are predicted to be inhibited. In orange, all the pathways are predicted to be activated. So the goal is to find different clusters, and in this cluster, we can find one particular set that these drugs are able to revert the activity of the corresponding pathways. So EPO might turn activated, CREB might turn inhibited. And these drugs are bortezomib, birabresib, and verurafenib. So just a quick view of what these drugs are. Bortezomib has been approved for myeloma, and it targets some component of the proteasome. The birabresib, it's a new drug which is in a clinical trial, not yet approved, multiple phase 1, some phase 1 and phase 2, target a component of the transport between the ER and the Golgi. So it's involved in transport. And the final drug is targeting the BRAF, and that has been approved multiple times in melanoma, metastatic melanoma. So the goal of using these analysis match is to scan through a set of 96,000 data set comparison expression and see at what point we can revert the pattern seen in our metastatic versus primary. And on that note, I will pass that to Tatiana, please.
All right, so in conclusion, I would like to summarize that we performed whole exome sequencing and RNA sequencing and used it for the integrated analysis of uLMS samples, which were collected over the course of the disease over time. So, so one of the results of this analysis is the detection of the 79 shared gene variants and 299 copy number variants. We also identified unique pathogenic variants, which were already known, such as TP53 and P10, and we also identified 6 other candidates. And one of the drivers of uLMS, in this case, we predict is the PI3 kinase AKT-mTOR signaling pathway, which is detected in about, based on the recent studies, about 30% of the uLMS. And activation of this pathway is known to play a role in multiple cancer-related processes, which lead to the cell growth and resistance to the chemotherapy, but not only that. There is a role of this pathway in development, and it might be very important in understanding of how to treat this disease specifically. And so, of course, we are looking for treatments, and we have hypothetic therapeutic drug treatments, as mentioned by Jean-Noel. And we're looking forward to do future studies on patient-derived cell lines and establish PDX models, validate our findings, and find molecular basis in more detail. And of course, we didn't show some of the data here. And we're looking for finding the right and the best treatment of uLMS. So, and with that, I would like to acknowledge many people who are involved in that project. from multiple institutions. So first I would like to thank surgeons from the Mount Sinai Hospital, Dr. Flores and Dr. Rahman, who performed the surgeries, which are state-of-the-art by itself, and the group at Memorial Sloan Kettering, where I was recently there, working as a senior scientist, Dr. Sam Singer and Dr. Will Tapp, both at the surgery department of MSKCC. And very special thanks to Dr. John Martinetti and his lab members who helped in moving this project forward, getting sequencing data, analyzing them, as well as establishment of the cell line. And there is also amazing work done by Dr. Boris Riva, who also doing computational analysis of this sample, which so this data were not presented here, as well as great financial support of LMS DR Foundation, headed by Sharon Anderson, and two also very important people, Janine DiTullio and Kelly Kimball, who are actively helping understanding this disease and how to find treatment. Thank you.
All right. Thank you, Dr. Omelchenko. Thank you as well to Mr. Pandya and Dr. Billaud for presenting your very important research. QIAGEN would also like to thank the audience for attending. If you have any questions about today's presentation, please email the address listed here or visit www.digitalinsights.qiagen.com for more information. Additionally, for a free trial of QCI Interpret Translational, the software discussed in today's presentation, please visit the URL listed here. Thank you again to all of our viewers, and have a lovely rest of your day. Until next time.